The overall goal of this procedure is to achieve spatially restricted gene expression in the mouse spinal cord by stereotactically injecting a viral vector into the lumbar dorsal horn. The first step of the procedure is to locate and expose the lumbar vertebra. The second step is to remove the dorsal portion of the vertebra in order to reveal the spinal cord, a procedure termed laminectomy. Next, a glass pipette connected to a microsyringe pump is positioned over the dorsal horn and lowered into the spinal cord. The final step is to slowly inject the vector suspension. 2 weeks later, transfection of dorsal horn neurons is demonstrated by the expression of green fluorescent protein, which allows tracking the virus's distribution.
This method helps elucidate the function of neurons in the somatosensory pathway. We use it in studies that focus specifically on the development of inflammatory or neuropathic pain.
Visual demonstration is critical for learning how to position the mouse into the stereotaxic frame. It is essential for the success of the procedure that the spine is fixed properly, because the laminectomy is such a delicate technique.
Martin Moll, a graduate student from my laboratory, will be demonstrating the procedure. John Wang, a research assistant, will be helping him.
One or 2 days prior to the procedure, pull the glass pipettes to obtain a tip diameter of 40 micrometers and bevel the tip at an angle of 20 degrees. On the day of the surgery, begin the procedure by disinfecting the surgical equipment and preparing the sterile surgical tools. To set up the stereotaxic frame, mount the microsyringe injector onto the manipulator and plug the device in. Attach one of